of the time on Thursday is going to be devoted to the second fun activity. Make sure you come to that. It counts basically as 5% of your grade, along with the other fun activity you did way early on in the course. It's going to be low stress, at least for me, it's going to be low stress.
Final?
The fun
Thursday.
on Thursday.
Make sure we get
No,
the reps on this.
well, the final is also going to be low stress for me, but yeah, I was referring to the fun activity. Show up. If you show up and participate at the fun activity, you're automatically going to get 80% on it. If you show up and manage to get a question right, I will give you 100%. If you show up and you get no questions right, but you're on the winning team, I'll give you 100% for that. If you show up and don't get 100 after all that stuff, then I will give you a 1 question or 2 question quiz to see if you can get 100% at the end of all that. Everybody's probably going to get 100%, so don't stress about it. And it will help, the fun activity will help prepare you for the final. So pay attention when I do it. You're welcome to record it when I go through it or whatever, but anyway, it's there to help you. So let me go through the final exam review then. I'm going to follow that study guide that's on Blackboard, and I think everybody has a copy. And if you have any questions, just stop me. I'll try to clarify it. Let me first just talk about the format of the exam. So basically, all the chapters we've covered so far in the class, except for 7, 8, and 11. All right, 7, 8, and 11 are not on the final.
Oh,
you don't have to
thank God.
worry about them. Okay, 7 and 8 were chapters that dealt with specific bones of the body. It was kind of like a hardcore anatomy chapter. There's no pictures that you have to worry about, so they're not on the final. And then chapter 11 dealt with kind of the specific muscles of the body, and for the same reason, it's not on the final either. But all the other chapters are. There's a total of 150 questions. And you should bring a number 2 pencil, because it's done with a Scantron. Those questions, some of them are going to be multiple choice, which you're used to. But then there's also going to be true, false, matching, and sequence questions. Now, you've done my true, false questions before, but these true, false questions on here are a little different. They're a pain in the butt. By the way, I did not write this final. This final has been in existence for a while. It's been used and then not used and brought back from the dead and used again and all this other stuff. But anyway, with the true, false questions, they always have 2 sentences. Every question has 2 sentences, and both sentences have to be exactly correct for you to say true. If any little detail in either of the 2 sentences is wrong, you need to mark the whole thing false. Okay? And sometimes those 2 sentences aren't even related to each other. Like one sentence might be about, homeostasis, and then the other sentence is about bones or something. They're unrelated, but just make sure the details are correct in both sentences before you say true. Otherwise, say false. Okay?
So even if they're unrelated, but they're
Even
true?
if they're unrelated, yeah. It's okay if they're unrelated, but they have to be true. Every detail has to be true about them. Yeah. So that's the true, false. Now, the matching questions, be careful with those. Those come in different sections. And make sure you read the instructions at the beginning of each section, because in some matching sections, it'll say that the answer choices can be used once, more than once, or not at all, right? Whereas in other matching sections, it'll say that the answer choices are only used once, which you can use to your advantage, right? If you're in one of those sections, you can kind of eliminate and kind of figure out where they go, right? So just pay attention to that. What else? The other thing to know about matching is that in some matching sections, there's some special ways that they get around the limits of the Scantron. Let me tell you what I mean by that. So you know how on the Scantron, you always have these 5 choices, right? Things like this. Okay? You have like the 5 choices. With the matching, suppose questions 1 through, I don't know, let's say 1 through 8. Suppose this was a matching section. Over on the left here, you have like the question or whatever, right? And then over on the right, you'd have a column of the choices that you could pick, right? Now, one of the limitations of the Scantron, as you guys know by now, is there's only these 5 choices, right? In some matching sections, they may have more than 5 choices, and the way that they get around that limitation, I'm sure some of you guys probably figured this out already or have seen it, is they start doubling up on letters. So if they had a 6th choice here, they would do something like, right? Or a 7th choice would be like this, right? An 8th choice would be like that. So for example, if you read this question and you wanted to pick this choice here, you'd have to color in the A and the B on the same line in order to choose it. Is that clear? Okay? You probably would have figured this out on your own next week, but you're gonna be stressed, you're gonna be like freaking out, you know, you see this, you're like, oh my God, what's this? You know, and they'll like come up and ask me like a billion questions, can I still be a nurse? You know, you'll be freaking out, right? You'll be all nervous. So that's that. The other type of question that comes up that you haven't seen yet are what are called sequence questions. Sequence questions are where you have to put things in order. They're basically like the matching sections, but they'll basically have the same number of choices as they'll have questions. So let's say you had 5 questions, then over here you'd have 5 choices. And you just have to put things in order that they happen. So for the first question to be like, what happens first? Then the second question, what happens second? What happens third? What happens fourth? And then what happens fifth? So, you know, you look at your choices over here, whichever one of these things you think happens first, you know, you'd put that over here and color it in. Maybe you thought D happened first. Whichever one you thought happened second, maybe it was A that happened first. Never third, maybe B, and then, you know, D, and then, or sorry, E, and then like C, something like that, right? So you're putting the things in order that they happen. And I'll show you today, I'll give you one specific example of some questions that'll be asked using the sequence method. And then on Thursday, I'll also give you some more examples of questions that they'll use like sequence on, so you should be in good shape. The problem with sequence questions is if you miss one, it can shift the whole thing and then you miss all the other ones, right? So just be careful with that when you do those sequence questions. That's about all I can say. Now, the good news about the final, well, 2 bits of good news. Number one, remember the deal that I made before that if you took all 4 lecture exams and you score higher on the final than you did on one of those 4 lecture exams, I will replace your lowest lecture exam score with whatever you got on the final, all right? I'll only replace one. So if you had 2 things that were tied for being the lowest, that doesn't mean I'll replace both of them with what you got on the final, okay? I'll only replace one. But anyway, that should be good news for a lot of you. And you know, don't psych yourself out. The final is not impossible. There will be people that get A's. There'll be a lot of you guys that will do better on the final than you did on one of your lecture exams. In past years, I'd say about 80% of students end up doing better on the final and end up replacing one of their lecture exam scores. So it's not impossible. Don't psych yourself out like, oh, this is gonna be miserable. All right, it probably won't be. It might be a little bit miserable, but it won't be like awful, all right? The other thing I want to tell you too is try not to be too worried about the time. Time is usually not an issue on the final. You've got 150 questions, which is just, you know, it's 50 more than your regular lecture exams, but you're getting the full classroom period to take it. You get the full 2 hours and 40 minutes to take it. And usually most students will finish and there'll still be an hour left, okay? You know, if you're that type of person that wants to wait, you know, and really double check to the end, yeah, by all means do that. I'm just saying that you probably won't feel the time crunch like you do on the lecture exams, okay? So really think about your answers, you know, go back to them, you know, all that kind of stuff. I think that's all I wanted to say about the format. Any questions about sort of the format or how it's going to work? The other thing too that makes it go a little bit faster is because there are a lot of these matching sections, there's not as much to read. When you have a lot of questions that are matching questions, there's less words involved than if you took the same number and had to write multiple choice questions for it, right? So it's less reading and that makes it go faster, okay? So something to be aware of. Now, in terms of the number of questions from each chapter, it's fairly even. It's about, I'd say about 11 questions, right about there for each. Some chapters will be a little bit more, some will be a little bit less, but it's kind of hovers right around 11, okay? The reason I'm telling you that is because in A&P2, when you get to A&P2 and take the A&P2 final, that's a very lopsided final. And that final has chapters where you'll have 30 questions from one chapter and then 2 questions from another chapter. It's very lopsided, okay? This one, it's not. It's very kind of even from each chapter. All right, that's kind of the overview. Any questions about like the overview or the big picture or anything like that? Everyone in pretty good shape? All right. The other thing I want to remind you too is don't forget, I did like about 3 or 4 weeks ago post the review video for the final on Blackboard. So you can watch that to your heart's content, maybe watch it once. I don't know, students like to listen to that kind of stuff, but that's a very passive process. Like, I don't really think, it may help you a little bit, but it's better if you just work on your own and try to actually do the hard work and study and make flashcards, whatever floats your boat. But if you want to like watch it or something, knock yourself out, or listen to it like you're in the car or something. All right, that's the big picture. Now let me go into each detail. I'm going to go through each chapter here in order, and stop me if you have any questions. I'm just following the final exam study guide, so that's what I'm doing. All right. So anyway, chapter number 1 was the introduction chapter. There are actually 10 questions from chapter 1. First, it says describe the anatomical position, metabolism, and homeostasis. What's the anatomical position? Yeah, it's when you're standing with your feet flat, your toes pointing forward, your arms straight, and your palms facing anteriorly, right? What's the definition of metabolism? When you eat something?
Wait, what did you say?
What's the definition of metabolism?
It's the combination of something.
It's the combination of something? She was on the right track. Alex had it. It's the sum of all chemical reactions in the body.
Yeah, some combination.
Okay. So it's all the reactions. And remember, the reactions in the body, there's thousands of different reactions, but they can basically be put into 2 groups, right? What's it called, the reactions when you build things? Anabolic reactions or anabolisms when you build things. What's it called when you break things down? Catabolic. Good. All right. And then homeostasis. What's the definition of homeostasis? Yeah, it's like balance or equilibrium in your different body parameters, right? You've got all these different body parameters: blood pressure, pH of your body, body temperature, blood glucose levels. All these things have to be kept within a tightly controlled range for you to survive. That's basically what homeostasis is. Okay. Next, it says describe negative and positive feedback and no examples. So, first of all, we're talking about what are called feedback systems, and with any feedback system, there's 3 parts. What are the 3 parts of a feedback system? Well, first, there's the part that monitors the situation. That's called the receptor, right? That then sends information into the second part, which is called the control center, or sometimes they call it the integrating center. That's where the information is going to get analyzed, and then if something needs to be done, a signal then gets sent out to the third part, which is called the effector. Okay, those are the 3 parts. Now those are the 3 parts, but then you can divide it into what are called negative and positive feedback systems. Both of them monitor different body parameters, but in the case of a negative feedback system, if a negative feedback system detects that a body parameter is outside of its normal range, what will the negative feedback system try to do? Bring it back or fix it, right? Try to reverse it, bring it back into that normal homeostatic range. So negative feedback systems always correct or fix the problem, however you want to say it. They help you to maintain homeostasis. What do positive feedback systems do when they detect like something's out of its normal range?
Push it further out.
Push it even further out. It's like they make the situation more severe. They take you away from homeostasis, right? Which are more common, negative or positive? Negative are a lot more common, right? They help you to maintain homeostasis. So you have negative feedback systems that control your blood pressure, that control your blood glucose levels, that control the temperature of your body, that control the pH of your body, all those things, right? What's the textbook example of a positive feedback system?
Childbirth.
Childbirth, right? Remember, with childbirth, you basically have the baby in there. The uterus is trying to squeeze the baby out. The baby's head is being pushed down, which stretches the cervix part of the uterus. There are stretch receptors in the cervix. They send a signal up to the mom's brain, and then the mom's brain instead of sending something down that corrects or fixes the problem, the mom's brain sends down a hormone called oxytocin, which makes the contractions even stronger. Stronger contractions, more stretch on the cervix, more release of oxytocin, stronger contractions, more, and it just keeps building and building and building. Because they take you out of homeostasis, positive feedback systems are potentially dangerous. So you always need some way to break the cycle. And how do you do it with the childbirth thing? Yeah, get the sucker out of there, right? They've been in there too long, get them out, and that'll break the cycle. Okay? All right. Know the levels of structural organization in order. These were the different levels you could study the body at. What's the smallest level you could study the body at? Smaller.
Chemical.
Chemical level is the smallest level. Then if you put a bunch of chemicals together, you get the next level, which is the cell level. Put a bunch of cells together, you get the next level, which is the tissue level. Put a bunch of tissues together, you then get the next level, which is the organ level. Put a bunch of organs together with a similar function, you then get the organ system. How many organ systems?
11.
11, and then put all 11 organ systems together, you get the organism, okay? Make sure you know that from small to big, like I just did, but also backwards from big to small, okay? And then pay attention when they ask the question that you answer it in the correct order, okay? Next, it says no common regional names, directional terms, planes, sections, cavities, membranes, regions, and quadrants. All right, so regional names, those were just fancy names for simple stuff. And there was that picture of that woman that had all those fancy names all drawn all over her body, right? What's the fancy name for like your neck area? Cervical. What's the fancy name for like your armpit area? Axillary. What was the fancy name for like your belly button area? Umbilical. What was the fancy name for your chin area? Mental, right? That's what they mean. So just kind of glance at that. Directional terms were terms used when you were comparing the relative position of two body parts. What's the directional term that means closer towards your head? Superior. And closer towards your feet is inferior. Closer to the back of your body is posterior. Closer to the front of your body is anterior. When you have two things that are on opposite sides of the body, you say they are contralateral. Two things that are on the same side of the body are ipsilateral. What's the term that means closer to the branch point? Proximal. And the one that's farther from the branch point? Distal. All right? Good. Planes and sections. So these were ways of kind of imaginarily dividing up the body into like different angles, right? What's the name of the plane that cuts like this, that cuts the body into superior and inferior parts? That's the transverse plane, or it's also called the horizontal plane, or it's also called the cross-sectional plane, okay? What's the name of the plane that divides the body into anterior and posterior parts like this? That's the frontal plane, also known as the coronal plane, right? I think somebody said it. And then the last major plane we discovered divides the body into left and right sides. What's that plane? Sagittal. It's also called the longitudinal plane. Now, if it divides it into perfectly equal left and right sides, it's called what plane? Mid-sagittal. If it divides it into slightly off-center sides, it's parasagittal, right? All right, good. Cavities. So there were two main body cavities, the dorsal body cavity and the ventral body cavity, but then there were subdivisions within each of those cavities. So for the dorsal body cavity, what are the two subdivisions of the dorsal body cavity? Cranial and spinal or vertebral, right? They use both names. What do you find inside the cranial cavity? Your brain. What do you find inside the vertebral cavity? Spinal cord, right? Then for the ventral body cavity, it's a little bit trickier. So what are the two main divisions of the ventral body cavity? Thoracic and abdominal pelvic. And what's the structure that divides the two? The diaphragm, right? All right, if you're in the thoracic body cavity, what's the name for the two cavities that are on the sides, like over here? Those are the pleural cavities. What organ is found inside the pleural cavities? Lungs. What's the name for the space in the middle of the thoracic cavity? Mediastinum. What's the name for the cavity that's inside the mediastinum? Pericardial cavity. And what's the organ inside the pericardial cavity? The heart, right? For the abdominal pelvic cavity, I guess it's a little simpler. What are the two divisions of the abdominal pelvic cavity? Abdominal and pelvic, right? Abdominal is mostly going to have digestive tract organs. Pelvic is going to have some digestive tract organs, but also like the bladder, for example, or in a woman like the uterus and things like that. Okay. All right. Regions and quadrants. Those were ways of dividing up the abdomen Remember with the quadrant method, how many quadrants are there?
4.
4, right? Right upper quadrant, left upper quadrant, right lower quadrant, left lower quadrant. With the region method, how many regions were there? 9 regions, right? All right. Be able to place, next section it says, be able to place organs into the correct body cavity, region, and quadrant. So I already kind of covered some of this stuff. In the thoracic body cavity, you have things like your heart, your lungs, they're probably the most famous. You also have your esophagus in the thoracic body cavity, your trachea, your thymus is in the thoracic body cavity, your aorta, your superior and inferior vena cava, they're in the thoracic body cavity. In the abdominal pelvic cavity, again, it's mostly digestive tract organs, except when you go down lower, like, again, the bladder would be there. The bladder kind of sits in the pelvic cavity, kind of in where that hypogastric region would be. If you're talking about the 9-region method, that would be where you find the bladder. Again, the fallopian tubes, the uterus, things like that would also be there. All right, that's all I want to say from chapter 1. Any questions about chapter 1? All right, chapter 2 then is the chemistry chapter, and it says first, describe the structure of the atom, atomic number, and valence electrons. So, what do you find in the middle of the atom? Nucleus. And what makes up the nucleus? Protons and neutrons. What kind of a charge do neutrons have? No charge, right? What kind of charge do protons have? Positive. And then what's the name of the little things that are kind of swirling around the nucleus? Electrons. What kind of charge do they have? Negative charge. All right. The atomic number. The atomic number specifically refers to the number of what? Protons. Okay? And that's also, you know, for that periodic table that's in the chemistry books, that's also how those elements are arranged on that table by their atomic number, right? Valence electrons. What are valence electrons?
The remaining electrons in the outer limit.
Yeah, they're the electrons in the outermost shell. In other words, they are the electrons that are farthest away from the nucleus. And they're important because they're involved in forming what? Bonds. All right, they're the guys that are involved in forming chemical bonds. And remember, for most of the elements in your body, there's a goal that they're trying to achieve. How many electrons do they want in their outermost shell? 8, and that was called the octet rule. The exception being hydrogen, which was satisfied when it had 2 electrons there. All right. Describe the types of chemical bonds, hydrophilic, hydrophobic. So there were 3 types of chemical bonds that we studied. What are the bonds called when you share electrons? Those are covalent bonds. And covalent bonds, if you remember, came in 2 types. If you share electrons equally, those are called non-polar covalent bonds. And if you share them unequally, they're called polar covalent bonds. When you have non-polar covalent bonds, no one ends up getting a charge. When you have polar covalent bonds, whatever atom is closer to the electrons is going to get a slightly negative charge, and whichever atom is farther away from the electrons gets a slightly positive charge. Okay. A good example of substances that have lots of non-polar covalent bonds are what? Water's a great example of a substance that has lots of polar covalent bonds. Remember, with water, the oxygen pulls more strongly on the electrons than the hydrogens do. So in water, the oxygen is slightly negative, the hydrogens are slightly positive. So water's a great example of polar covalent bonds. But what's a good example of non-polar covalent bonds? Lipids and fats, right? They have lots of non-polar covalent bonds, and they usually don't have any charge to them because of that. Okay. Next type of bond we talked about were when you transfer an electron from one atom to another atom. What type of bonds were those called, where you transfer an electron? Those are ionic bonds, okay? And the example we gave was sodium chloride, like your standard table salt, right? Sodium gives up its electron, which causes sodium to have what kind of a charge if it loses an electron? Positive. Chloride gains the electron, which causes it to have a negative charge, and that difference in positivity and negativity draws them together, and that's an ionic bond. And then the third type of bond we talked about were hydrogen bonds. That's where you have a slightly positive hydrogen on one molecule forming a bond with a slightly negative atom on a different molecule, usually a slightly negative oxygen, for example. Some places where we saw hydrogen bonds, 3 places were in proteins, helping to give like the secondary structure of proteins. Between water molecules, when you have like a whole bunch of water, like a glass of water, holding water molecules together, and then also in between the DNA strands, holding the 2 strands together, right? Hydrophilic. What does the word hydrophilic mean?
Water loving.
Like water loving, right? That just means substances that dissolve in water, okay? They love water because they dissolve in water. Substances that dissolve in water usually do so because they have polar covalent bonds, okay? When a substance has a polar covalent bond, it gives it a charge, just like water does, and if you follow the chemistry principle of like dissolves like, things that have a charge will dissolve in water because water has a charge, okay? What does hydrophobic mean? Water fearing, right? It doesn't dissolve in water, usually because it doesn't have a charge, usually because it has lots of non-polar covalent bonds. And you know, if you go through your 4, and we're going to do this in a second, go through the 4 major groups of organic compounds. And what are the 4 major groups of organic compounds in the body? Carbs, lipids, proteins, and nucleic acids. Of those 4, the carbs, the proteins, and the nucleic acids, they're usually hydrophilic. They usually dissolve in water. Lipids are the oddball. They're usually hydrophobic. They don't dissolve in water. Okay. Next it says, what would influence the rate of a chemical reaction? In other words, what could make like a chemical reaction go faster? And there were 3 things we mentioned.
Temperature.
So temperature's 1. When you heat things up, that makes the reaction go faster. You said catalysts, right? And what's the name we have for catalysts in our body? Enzymes, right? Enzymes are chemicals that specifically speed up other chemical reactions, right? And then one more thing that can speed up a reaction. The amount of the reactants, the concentration of the reactants, right? If you have a jar and you put just a little bit of substance A in and just a little bit of substance B in, those would be like low concentrations, and it'll be a while before they even find each other. Versus if you took that same jar and put a whole bunch of A in, a whole bunch of B in, those are high concentrations. Just the likelihood that they find each other and start to interact is higher, right? So it speeds up the reaction. All right, describe the pH scale in terms of the amounts of hydrogen and hydroxide ions. So what's the range of the pH scale? It goes from what to what?
0 to 14.
0 to 14. If you're down at 0, you're really what?
Acidic.
Acidic. And if you're up at 14, you're really basic. Okay? The closer you go down to 0, the more you have of the hydrogen ions. Okay? The closer you go up to 14, the more you have of the hydroxide ions. Okay? Next it says, describe the 4 groups of organic compounds, saturated versus unsaturated, levels of structural organization of a protein, enzymes, coenzymes, and ATP. All right, so a lot to go through here. So first of all, you already mentioned the 4 groups of organic compounds. Those are the carbohydrates, the lipids, the proteins, and the nucleic acids. So let me go through each of these in a little bit more detail. First of all, what does it take to even be considered organic? You need to have carbon, okay? Now sometimes they'll modify that definition and say that the carbon also has to be bonded to at least one hydrogen or to another carbon. And the reason why they sometimes do that modification is because they want to exclude carbon dioxide. Most scientists don't consider carbon dioxide to be organic, right? So they sometimes use that definition. All right, let's talk about the carbohydrates first. What does your body like to use carbohydrates for?
Energy.
Energy. It's your body's preferred energy source, especially glucose. Glucose is your body's favorite carb for energy. Now, when you've eaten and when you have extra glucose left over, your body just doesn't store the glucose as glucose. Instead, it converts it into what to store it? It turns it into glycogen in order to store it. Glycogen is a big polymer of glucoses. It's a whole bunch of glucoses that have been combined. And what are the 2 main organs or types of tissue in the body that store lots of glycogen? The liver and skeletal muscle. Liver and skeletal muscle are the main place. Okay. Now, your body loves to use carbs for energy, but there are also some other jobs for carbs. For example, one important job of carbs is to serve as cell recognition markers on the surface of your body cells, okay? Everybody's body cells will have some carbs on the surface that are unique for their own body cells. So the pattern of carbohydrates on a cell's surface sort of acts like a name tag for that cell, and it's there to mostly help your white blood cells to be able to identify your own body cells from like a foreign invader or someone else's body cells, okay? And they call that like cell recognition markers or self-recognition markers, and it's really due to the unique pattern of carbohydrates that we all have on the surface of our body cells, okay? We'll get more into that topic in AMP2 when we do the immune system, but it's just something to mention now. Next group are the lipids. Remember, the lipids are kind of an oddball because they're usually hydrophobic. Will your body use lipids for an energy source? Can it use lipids for energy?
Yes.
Absolutely. Yeah, your body can certainly use fat for an energy source. Unfortunately for us, if you're trying to lose weight, your body always likes to go through the carbs first before it really starts attacking your fat, right? But yes, lipids actually are a good source of energy. But there are also other jobs for lipids besides being an energy source. For example, what's the main component of your cell membranes? The blankety blank blank blank.
Lipid.
The phospholipid bilayer, right? Phospholipids are a type of lipid, right? There's an example of a lipid not just being used as an energy source, but it's there, for structural purposes. Obviously, that's a huge deal. The phospholipid bilayer, it's on all your body cells. It's super important. So that's a good example. Another good example would be like cholesterol. All right, everyone's always hating on cholesterol, but cholesterol does a couple of things. First of all, it's the basis for making certain steroid hormones in your body, like aldosterone or estrogen or testosterone. They're all based on cholesterol, okay? Cholesterol is also important in your cell membranes to kind of stabilize them and prevent them from breaking apart, okay? The most common dietary fat that you have, and the one that gets stored in your fat cells, is what lipid? The triglyceride, okay? The triglyceride is the main dietary lipid. It's the one that gets stored in your fat cells. When you look at triglyceride, it's made up of a glycerol backbone, and then there are 3 fatty acid chains that are attached to that. And you got to look at those fatty acid chains carefully for the bonds that are between the carbons in the chain. If you find only single bonds between the carbons, what kind of a fatty acid is that? That's called a saturated fatty acid. So that would be like my diet. Sorry, I didn't do new diagrams for you guys. I'm too lazy. But anyway, this would be like up at the top. This would be what a saturated fatty acid looks like. It's only got single bonds between the carbons. What's it called then if you have a fatty acid that looks like this, where you have some double bonds between the carbons? That's unsaturated. Okay? All right. Next we have proteins. Does your body like to use proteins as an energy source?
No.
No, it hates to. All right? That's because there are way too many other important jobs for proteins than to just simply break them down for energy. Okay? For example, almost all of the enzymes in your body are proteins. Okay? And enzymes are super important. Okay? Proteins give strength to your body. Right? For example, the number one most common protein in your body is this structural protein that's called collagen. It's like a tough rope, right? Proteins, are needed for muscles to contract, right? Remember the myosin and actin that need to work when your muscles contract. Proteins can serve as carriers. For example, hemoglobin, which carries oxygen around your body. That's a protein, okay? So many important jobs for proteins. They're also the most complex of your body molecules. That's because they're made. What are the building blocks for proteins?
Amino acids.
Amino acids. How many different amino acids? 20. And when you build a protein, you can use as many of those different amino acids as you want. They can be in any order. You don't have to use them all. You can have repeats if you want. So that leads to a lot of complexity. And in the next chapter, we talk about protein synthesis, which is how you make the proteins, and it's a pretty complicated process. Okay. What's the name for the type of bonds that you have between the amino acids? Anyone remember the name of those bonds? They are called between amino acid bonds. No, there's something cooler than that. There's a better name. What are they called? Peptide bonds. Yeah. So peptide bonds are in between the amino acids. The type of reaction that pulls those amino acids together, it's called a dehydration synthesis reaction. You're basically removing water from the equation, and then that forces the amino acids together. With proteins, it's all about their three-dimensional shape. Proteins have to have the right 3D shape in order for them to function. And for most proteins, the 3D shape is based on what's called their tertiary structure. Okay? If you remember from this chapter, there are 4 kind of levels that you can study proteins at. There's the primary level, the secondary level, the tertiary level, and then the quaternary level. The primary level for studying proteins would be just if you took a protein and you just wrote out the amino acids in order. You're not really even trying to show the shape, you're just listing the amino acids in order. The secondary structure is starting to show some of the shapes, the twists and the turns, but it's not showing you the full 3D shape yet. The tertiary structure is usually the highest structure that you can go to for most proteins. The tertiary structure is showing you the entire 3-dimensional shape of one polypeptide chain. And the reason why I say that's the highest level is because most of the proteins in your body are just made of one polypeptide chain, so you can't go any higher. So for most proteins, the 3D structure of the protein, in other words, the tertiary structure, is what determines its function. There is one more level, though, the quaternary level, and that's reserved for really, really big proteins that are made up of 2 or more polypeptide chains, such as hemoglobin. All right? Hemoglobin's made up of 4 polypeptide chains. So if you just wanted to look at one of the 4 for hemoglobin, you could go to the tertiary level. But if you want to look at the entire hemoglobin with all 4, then you have to go to the quaternary level. Okay? Last, we have the nucleic acids. What are the 2 nucleic acids you have in your body? DNA and RNA, right? DNA and RNA are your 2 nucleic acids. Where do you find DNA in your body, in your cells, I mean? Inside the nucleus is where you find most of the DNA. And then remember, the DNA inside your nucleus is organized into these 46 chromosomes, right? However, and I heard Jess say it, there is one other place where you can find DNA in your cells, and that's inside of mitochondria. And the funny thing about that DNA is it always comes from where? Your mom, okay? DNA, the structure of DNA, what's the name for that special twisting structure they have for the DNA? The double helix, right? Remember what are the 4 bases that you have with your DNA? You can just give me the letters. A, T, G, and C, right? A, T, G, and C, that's for DNA. And remember, there are certain what are called base pairing rules. The A always binds to the T, and the G always binds to the C. Remember that? All right, so that's DNA. RNA, some differences. Remember, RNA, you can find it either in the nucleus, or you can find it out in the cytoplasm. RNA comes in 3 different forms. You can have messenger RNA, you can have transfer RNA, or you can have ribosomal RNA. Also, there's a difference between them in their structure. Remember, DNA is double-stranded, RNA is usually single-stranded. And then the difference that students usually remember is the fact that they have different bases. What's the different base that you have in RNA that you don't have in DNA? Yeah, the U, which is uracil. So instead of the thymine, the T, you have uracil in the RNA. Okay. One other difference too, sorry, I just remembered it. The other difference is in the 5-carbon sugar that they have. Remember, deoxyribose is the sugar that you have in DNA, whereas just ribose is the sugar that you have in RNA, right? Next it says enzymes. Again, enzymes are substances that speed up chemical reactions. Usually they're going to be proteins. Usually they will hold the reactants in such a way that less energy is needed to make the reaction happen, and that's going to speed up the reaction, okay? You've got thousands of different chemical reactions in your body, and each one needs its own specific enzyme to make it happen fast enough so that you can survive, okay? Coenzymes are substances that sometimes are required to attach to enzymes in order to turn them on. Coenzymes often come from vitamins, okay? ATP, what does ATP stand for? Adenosine triphosphate. And what does your body use ATP for?
Energy.
Energy. It's kind of like the energy money of your body cells. Anytime you have something that requires energy, one of the big ways that you pay for it is by basically spending ATP, okay? All right, that's all I wanted to say about chapter 2. Any questions about chapter 2?
I'm gonna have to know how to find out that changing AUG, any of that.
The change, what do you mean?
But when you change with the translation, DNA translation.
Oh yeah, but that comes up in the next chapter.
Oh.
Yeah, you almost got it, yeah, but not quite. All right, let me go on to the next chapter. This is hopefully, I'll try to get done with this. This is probably the last chapter I'm going to cover today. Everything else I'll have to cover on Thursday. Again, if you want to, watch the review video. It is posted, all the stuff's covered there, but anyway. Chapter 3 is the cell chapter. There's actually 7 questions from the cell chapter. First it says, describe the structure and function of a typical cell membrane. So first of all, again, what's the main component of any cell membrane? The blankety blank blank blank?
Phospholipid
The
bilayer.
phospholipid bilayer, right? That's the main component of a cell membrane. And remember, kind of floating in that phospholipid bilayer, you have these proteins that have different functions. Remember that the plasma membrane is considered to be, it has selective control over what moves in and what moves out of a cell, okay? It doesn't just allow everything in or everything out, it actually controls what goes in and what goes out. Next it says differentiate between passive and active transport and no examples of each. So with passive transport, with both methods, you're talking about moving things across cell membranes. With passive transport, what don't you need to make it happen? You don't need any energy for passive transport, okay? That's because passive transport is always based on the natural process of what? Diffusion. Diffusion is the natural tendency for a substance to move from where it's in high concentration to where it's in low concentration. And so long as you're going across the membrane in that direction, from high concentration to low concentration, that's natural, and you don't need any energy to happen to do that. So all of the passive processes are based on diffusion. And there were 3 types of passive processes that we mentioned. Simple diffusion is when you have something that's small enough that it can pass straight through the phospholipid bilayer. Facilitated diffusion is when you have something that needs a little help to get through the cell membrane. It's still diffusion, but it needs some help, either in the form of like an ion channel or in the form of a carrier or something like that. And then the 3rd type of passive transport was osmosis. That refers to the diffusion of what? Water, all right? So those were all passive. Then you have active transport methods. Active transport means you need what to do it? Energy. The reason why you need energy is because with active transport, you're trying to go against diffusion. You're trying to move a substance from the side of the membrane where it's in low concentration, and you're trying to move it to the side where it's in high. There's already a lot there. So if you want to do that, that's not natural. And to make that happen, you're going to have energy to make it happen. There were 3 types of this. Primary active transport means you're using ATP as the energy source. A good example of that was the sodium-potassium pump, right? Secondary active transport is when you move 2 things at the same time. One thing moves naturally following diffusion, and the energy of that thing moving naturally is used to provide the energy needed to make a different substance move against its concentration gradient. And if the 2 things are moving in the same direction across the membrane, that's called a symporter. If the 2 are moving in opposite directions across the membrane, that's called an antiporter. And then the 3rd type of active transport was transport in vesicles. That's when you usually have things that are fairly big. You put them into these sacs that are called vesicles, and you can use that to either bring stuff into the cell or move stuff out of the cell or move across the cell if you want to. What's it called when you bring something into the cell using a vesicle?
Endocytosis.
That's endocytosis. And there were 3 types to that. There's receptor-mediated endocytosis, phagocytosis, and then pinocytosis, okay? What's it called then when you move something out of the cell? Exocytosis. And then what's it called when you do a combination of both, where you do endocytosis on one end and then the vesicle kind of goes across the cell, and then you do exocytosis on the other? That's transcytosis, okay? All right. Next it says, describe hypo, iso, and hypertonic. So these are words that are used to apply to different solutions based on how concentrated that solution is compared with the insides of the cells that are in that solution. And if the solution is more concentrated than the inside of the cells are, you say that's a hypertonic solution. If the solution is less concentrated than the insides of the cells are, then you say it's a hypotonic solution. And if the solution has the same concentration as the insides of the cells do, you say it's an isotonic solution. Okay? All right, next it says sequence the events of protein synthesis. And this is going to be one of those sequence sections that I was talking to you about where you're going to have to put things in order. Now, due to just sheer laziness on my part, again, I apologize. I did not rewrite this for you guys, but let me, well, you know what? Before I show you the thing, let me just talk you through it, and then I'll show it to you, and you can take a picture of it if you want. But anyway, protein synthesis is the way that you make proteins. Proteins are complicated because they can be made up of 20 different amino acids. You can put those in any order. You don't have to use them all. You can use repeats, so it's very complicated. And because it's so complicated, you have to have a plan or a blueprint for building each of your proteins. And the blueprints for building your proteins are basically your genes. Okay, your genes are segments of your DNA inside the nucleus that are on your chromosomes. So, you know, if you have a particular protein that you want to make, you first have to go into the nucleus. You got to find the chromosome that has that gene on it, and if you take that chromosome and you basically unravel it, the chromosome will be a really long piece of DNA, and then someplace on that chromosome there should be the gene for that particular protein that you want to build. Once you find the gene for the protein that you want to make, then you go through protein synthesis, and there are two T words that are associated with protein synthesis. What are the two T words associated with protein synthesis? Transcription and translation, right? Where does transcription happen in the cell? Always inside the nucleus. And during transcription, you're starting with that gene, which is made out of DNA, and you're using it to make what? Messenger RNA. Okay? That's transcription. Then you do translation. Where does translation happen?
Ribosome.
That happens in the ribosomes, which are out in the cytoplasm. And so during translation, you're taking the messenger RNA, you're feeding it through a ribosome, and then that's being used to make the final protein. Okay? So those are the two processes. Now let me go in a little bit more detail through each of these. Okay? So first of all, let's start off with the transcription part. You're inside the nucleus, you have found the gene for the protein that you want to make, and there's an enzyme that will attach to that gene and start this process of basically building the messenger RNA. What's the name of that enzyme that makes the RNA? You're close. Yeah, it's the RNA polymerase is the enzyme that starts the transcription. It basically attaches to a little spot just before the gene known as the promoter region. And then the RNA polymerase slowly moves across the gene, reading the DNA. And as it does, it calls for matching RNA bases to be brought up, and you build this thing called messenger RNA. Now, that messenger RNA that you first get, is it immediately ready to leave the nucleus?
No.
No. What did you have to do to that messenger RNA first before it could leave the nucleus? You had to splice it, which basically means cutting out the pieces that you don't want and then gluing together the pieces that you do want. What was the name for the pieces that you don't want?
Introns.
Introns. I know I always felt like these names sounded like they were backwards. And then what's the name for the pieces that you want to keep? Exons. So you cut out the introns, you glue the exons together, and then you have your final version of the mRNA, and that's going to then leave the nucleus. The mRNA leaves the nucleus, and then it goes out and finds a ribosome. And the ribosome basically attaches the mRNA, and it's going to start the process of translation. Okay? How does the ribosome know where to, or where does it attach on the messenger RNA?
To the starting point.
To the start codon, right? And the start codon was always what three letters? AUG, right? So that tells the ribosome, hey, this is where you want to start translation. So the ribosome attaches there, and then slowly the ribosome works its way down the messenger RNA, essentially reading the different codons as it goes along. And as it reads a codon, it calls for a transfer RNA to bring a different amino acid up into place, and the amino acids get attached to each other via dehydration synthesis reactions. So you create this growing chain of amino acids, right? Eventually, though, you have to finish the process. And how does the ribosome know when to finish things?
Stop codon.
It comes to a stop codon that tells it, okay, you're done, and then you have your finished protein. Okay? So that was me talking you through it, and now I will reveal what I wrote for the other class so you can just look at it. It's basically what I just went through, but here it is in all its glory. I will sign it for extra money. No, I won't sign it. Okay? And that's where I'm going to stop for today. Sorry, one more topic. I apologize. Sequence the events of cell replication. So, sorry, this refers to mitosis and cell division. What's the first phase of mitosis?
Interphase.
Prophase.
Prophase.
All right, so during prophase, 3 things are happening during prophase. Number 1, you see the chromosomes for the first time. Remember, before prophase, you're in this thing called interphase, and the chromosomes are kind of unraveled. You really can't see them. They're there, you just can't see them. But during prophase, they condense. You see the chromosomes. Number 2, the nuclear envelope that surrounded the chromosomes, that disappears. And number 3, you build this thing that kind of looks like a football that's called the spindle, the mitotic spindle. All right? That all happens during prophase. What phase happens after prophase? Metaphase. What happens to the chromosomes during metaphase? They all line up single file in the middle of that spindle. What comes after metaphase? Anaphase. Anaphase, again, starts with the same letter as the word apart. That's when the chromosomes get pulled apart, and they get pulled to different ends of the cell. And then what's the last phase? Telophase. The chromosomes now are really far apart from each other, and now a nuclear envelope will reform around the 2 new sets of chromosomes. Okay? At the same time that anaphase and telophase are happening, the cell membrane is also working its way in to actually divide up the cell into 2 separate cells. What's that process called where you're physically dividing it into 2 separate cells?
Cytokinesis.
That's cytokinesis, and it's overlapping with anaphase and telophase. Okay? Yes?
Are we gonna have to know all 3 stop codons?
No. No, you don't. But you do need to know the 1 start codon. Yeah. Yep. All right. I mean, at this time, that's where I'm going to stop today. Sorry about the false alarm. Any questions about? Yes.
For the passive transport, do we also know the simple diffusion, channel mediated diffusion, and carrier mediated?
Yeah, you should be aware. I mean, those are all subtypes of passive diffusion. Yeah, you should be aware of them. Yep.